Hi everyone, my name is Cilia. I'm the founder and CEO of Cilia. So today I'm going to introduce you one new technology called microscoop. Microscoop is of course, it's like scoop is like ice cream scoop. You isolate the protein, kind of picking them up. So this talk is more about applications. So different kind of problems you can study using microscope. It's really broad, from tissues, cells, basic sciences, clinical related, drug discovery related. So I will give you a few examples. Here, the background is actually one of the studies we got the proteome list. And as you will see, it's pretty high sensitivity and specificity of this method. So really, there are many biological problems. I mean, you see from the eyes that they have specific locations that you know they are different, or you know they are like interface or interactions. But it's very hard to isolate the protein in such small location or in high abundance, right? So in the neuroscience, cell biology, cancer biology, metabolic, or many other fields like immunology, that this image happen again and again in pathology, that people see the image, they know, okay, there are diseases, but you don't exactly know what exactly in this triple negative breast cancer, for example. That's this method is going to enable. So the system is like this. There is a microscope in the middle, and we designed this optical system so that you can do this spatial protein discovery by this very high speed coordination of all this. If you want to know more about the methodology, we have another video talking about methodology in more one step by step, so you can see that video. But here I want to quickly go through this methodology and then give you more application examples. In the methodology side, you basically in the media you put this photoactivatable bouton probe. So within this probe, within this sample, this probe, once you shine light, protein will be biotinylated. So you have to determine the location you're interested in. In the lower left corner, you can see if you are interested in this red compound. So you can define by AI or image processing. So AI is like you train the computer to understand what you are interested in. So you can train on the side with GPU, maybe with 100 images. Then later on, all these thousands or millions of images, they can recognize automatically the location you're interested in. And then with the location you're interested in, you shine light to this location. We use a 2-photon light to trigger the photo-induced biotinylation. So say if each panta has 300 different kind of proteins, then this 300 different protein will be biotinylated. So we do this again and again for many field of view, actually sometimes 10,000 field of view or more. Then you pull enough protein, because so far we don't have PCR for proteins, right? So you really need to beat the protein amplification problem here by protein accumulation. That's why so far no other method can do it, because everyone is kind of thinking, how do you get enough protein for detection, unbiased detection, like using mass spec. So once you have enough protein biotinylated, say in this panta, maybe millions of this panta, then we do protein extraction by surveillance beads, and then you become the spatial purification, right? You purify the protein, very specific the location you biotinylate, and then you send all these purified biotinylated proteins into the mass spec to determine what they are. This is a real experiment for stress granule. So stress granule are, you can see they are very small inside the cell, maybe between 500 nanometer to 3 micron size. So to pick them up is very hard. So now you can see this is automatic photobiotinylation. You recognize on the fly. So imaging, 0.1 second, image processing or AI calculation, 0.1 to 0.5 second, and you shoot live in this location. So this is really like an AI video game. So you shoot down all the enemy, you know where all the enemies are, and you just calculate where they are and then do the photo-induced biotinylation. And each contact inside, there are 300, 500 different proteins, will be biotinylated, and you can pull them out to determine what they are. I'm going to give you 6 examples in this talk pretty quickly. And we do many more problems, including many other FFPE tissues, human brain, other kind of organs. Those are more other people's data, so we don't show it here. I'm going to show you these examples, hopefully to show that the kind of universal power of this method for proteomic discovery. Cilia is one example. It's the original research I studied that kind of motivated me to do this when I was at Columbia. You can see we can biotinylate the protein inside the cilia very precisely. And then once you biotinylate it, you can pull them out later. Right? So what we did was we have one sample without light and one sample with light. And this photo-labeled protein in the cilia, then you do both samples using mass spec to get a proteome for both. And then what we did was we take the ratio of these 2 samples for each different proteins, and then the copy number, more copies in the photo label are those like enriched in the photo label, right? So you have a kind of some protein that still showing low copy in the unlabeled one, but the one the labeled will be increased significantly. So as you can see, this volcano plot on the right side, more of the enriched proteins by light. You can see the x-axis is the ratio of the photo label versus unlabeled. Most of the cilia protein is situated in this right side. So showing that not only high sensitivity, it's also high specificity. So the end result is one long list of proteome. You can see all these proteins. And as a cilia researcher, you can see many IFP proteins here, many WDR proteins here. So it's very good in specificity and sensitivity. And you can even discover some proteins like people, maybe they study on their own, and then we really can discover here that maybe no one showed before. If you map to these rectome pathways, you can see that many of the pathways exactly what the primary cilia is involved. If you zoom in more, you can see in the middle intraflagellary transport, on the right autophagy. So these are all known pathways that related to primary cilia. And microscope kind of recover all these pathways by this light-induced biotinylation way. And then this has become very precise, right, on many problems that can be studied using this method. The more important thing is we discover a new protein constituents in primary cilia that people didn't study before. Here are just a list of the top 30. And then we can even zoom in further to see what they are involved. You can see on the lower left corner, this signal transduction and endosomal related proteins that previously didn't discover too much. Then now with this study, we find many more. Intraflagellary transport, you can see that cluster, we discovered no new ones because there's been detailed, careful study, transfusion zone as well. We basically almost discovered none of the new ones. But the new ones that's the most interesting for people to go further that people probably couldn't do it with other technology. So this is the second example, the stress granule. So we isolate the protein in the G3BP1, that's the marker for stress granule. And you can see the biotinylation is very precise. And the right side is 3 repeats. Again, you get a long list of the related, the isolated stress granule protein. And then for the stress granule researcher, you can see, okay, this is really specific. And then again, mapped to the reactome pathway, also recover the pathway related to stress granule. But the interesting thing is the newly found protein. So here, the green ones are the protein that people already know using other studies, including the approximate labeling like FX2. But you can see we've discovered several ones, like the gray one that people didn't know, like PD, IMO7, all these proteins that people didn't know that belong to stress granule. So how do we know we are non-specific or we discover new ones? Then the best way, the first trial is to see if they colocalize in the stress granule. So what we did is unbiasedly pour all the antibodies for the top, and then to see if they localize in the stress granule location. Here is just one example, RPSA. You can see they colocalize very well with BP1 in the stress granule location, showing that the discovery power of this method. You can find the protein that people never study related to stress granule, and they will colocalize it. This is another example, PPIA. You can see that the stress granule, not only they colocalize, but you can see on the right side, kind of there are two kinds of stress granule. One is two color, one is one color. So we really finding new biology. You can use these to study further to see what the difference of these two kinds of stress granules. So here are 12 examples that we find a new protein constituents in the stress granule site. So these are all, you can see them colocalize very well with BP1 in the stress granulation. So you are not only discovering one or two, you discover a bunch of proteins that they colocalize very well. So after you counting all this, the specificity together is above 90%. What we got. This is another example, a T-cell B-cell interface. In this example, we use AI to recognize the location of a T-cell B-cell interface. And you can see we really recover many known immune synapse related protein, and also some protein in the B-cell side, some protein in the T-cell side. So this showing that we really can do this for many different problems, even as small as immune synapse. We discover a new immune synapse proteins. This can be further studied by the immune synapse researchers. Then, of course, there are many different kinds of immune synapse, alpha beta T, gamma delta T, or different involvement in the cancer immune therapy. Okay, this is another example for Alzheimer's. So we study the protein in the beta-amyloid aggregates or plaques. So what we use here is a cell model, use SY5Y cells, and we differentiate them to neuron-like cells and overexpress A-beta 142 to show where the granules are. And we only kind of scoop the protein in this granule site. And you can see we really recover many known A-beta associated proteins. And not only recover this, we find the new ones. These are two new one examples that the long protease and DDX3X helicase. You can see they also colocalize very well with beta amyloids. So this is really not only the basic science now, it's really related to disease. And we finding these new proteins, we are studying their roles in the mouse model. Another example here, Mossy fiber bouton. This is a pretty challenging project using the fresh frozen mouse hippocampus. The challenge is because that very few of these boutons in the brain. You can see the green part is this bouton site. And then we biotinylated this protein very precisely. And every sample maybe only 50 of these, very few. But even with this few, you can isolate the protein, collect all of them together, and then identify what they are. And you can see we have a big noise peak on the left side, but on the right side, this second peak are the protein that's new. So here you can see the x-axis is the ratio between the photo label versus unlabeled. So we take the ratio of these two to see which protein are enriched. And then the enrichment protein, many of them that we know are neuron growth related, and we find many new ones that is worth the further study. So we did this for 3-month-old mouse, 6-month-old mouse, and then compare the proteome change related to our neuron growth in different age. The next example here is more really toward a very small area, this lipid droplet mitochondria interface. So this kind of organelle interface is super tiny location. And this problem, of course, is related to fatty liver. So we treat these cells, the liver cells, with kind of oil, right? So the fatty acid, in this case, oleic acid, and then trying to see when the lipid droplet aggregates, what proteins are related to this metabolism in the mitochondria site. So what we did is we generate a mask using lipid droplet, and then we generate another mask with the mitochondria. And you take the intersection of these two, and then we only zap the protein in this intersection. I mean, it's not very precise, right? You can see that some of them may not be really the interface because of the signal. But when we zap so many of them, we zap maybe 1 million of these locations. And then the protein really there will be accumulated 1 million times, and they are not there, will be averaged out, right? So you really identify the protein in this peridroplet mitochondrial interface. And then, as you can see, we really recover many of the lipid droplet proteins. And if you see the protein we find, these are protein A, we say here, you really can see this new phenomenon, right? So the protein, without this oleic acid treatment, this green protein, they don't colocalize in this lipid droplet. But once you treat with oleic acid, they kind of wrapping around this oil. That kind of people never seen this before. You really identify the protein, very small area that surrounding this oil. And we identify several of them, not only the wrapping around one, also the one that sporadic in the mitochondria interface. This is another protein, another example. You can see this one also, you treat with oleic acid. They really localize this surrounding part of the oil. So now it becomes a possible target for fatty liver, right? So if you treat this protein, these newly identified protein constituents surrounding the oil, maybe you can dissolve the oil or do something damage this oil. Okay, so with this 6 example, I hope I show you that the microscope is really this high precision spatial protein purification method. So this is the method that didn't exist before. And with this ability now, it facilitates many problems that can do unbiased subcellular spatial proteomic discovery. So the proteomic discovery now, because of this highly multiplexed, many different field of view, so you can get the high specificity and high sensitivity. And it can be applied to cells, tissues, even 5-year-old, 10 years old FFPE tissues. We can do it. And this has been used for many studies, primary cilia, stress granule, or other problems I just showed. So you can imagine many studies can be done with image-based, image-guided proteomics, right? So now it opened up a lot of opportunity for researchers in different fields. Thank you very much.